Okay, so yeah, there was a little internet connection issue last time. We have understood the lack operon, and now we go into the basis of the genetic code, everything about protein synthesis and how the rules of protein synthesis are laid out. Let's understand the concept of protein synthesis, the logics behind it. What kind of information do genes have? Genes में क्या information होता है? Beadle and Tatum explain the concept of information of genes. They expose spores of the fungus called Neurospora crassa. Remember, RF sir had taught you Neurospora crassa is used for studying mutagenic studies. Sir, spores of fungus Neurospora crassa were exposed to mutagenic agents like X-rays. So, spores of the fungus Neurospora crassa were exposed to X-rays. X-ray exposure किया गया and that caused a mutation in the DNA. तो डीएनए में म्यूटेशन हुआ and this mutant spore could not produce enzyme. This mutant spore, Neurospora crassa, lacks certain enzymes. And what was the enzyme for? Neurospora crassa is a fungus. Fungi are saprophytic. Fungi are saprophytic. They give out enzymes which help in digestion and formation of new spores. तो सर फंगाई सैप्रोफाइटिक होते हैं, उनमें से एंजाइम्स निकलते हैं and those digestive enzymes help in replication of the fungi to form new spores. When this spore was exposed to X-ray, the gene undergoing formation of the saprophytic enzyme underwent a mutation. तो mutate हो गया and the digestive enzyme was not produced. Digestive enzyme was not produced and replication of spores stopped. So that clearly meant that one gene has undergone a mutation. It concluded that a gene is a segment of DNA which codes for a specific enzyme. And if the enzyme is mutated, the gene is mutated, the enzyme will not be produced. That was called as the one gene, one enzyme hypothesis. One gene, one enzyme hypothesis was given by Beadle and Tatum. ये याद रखना नीट में बहुत बार पूछा गया है. Who has given the one gene, one enzyme hypothesis? Beadle and Tatum. But later on, scientists proved it that every enzyme is a protein, but every protein is not necessarily an enzyme. Enzymes are chemically proteins, but proteins are always not enzymes. So, one gene produces one enzyme is wrong. It can produce a protein which is not an enzyme. And in 1957, this hypothesis was modified by Vernon Ingram as one gene, one protein hypothesis. By Vernon Ingram, one gene as one protein or one polypeptide hypothesis. One gene, one protein or one polypeptide hypothesis was suggested by Vernon Ingram. Later on, the genetic code was suggested. What does the genetic code know? We know all the 8 points. DNA contains all genetic information of the organisms. And it is the genes which control, regulate, and express the characters. A gene found in the DNA will express the character. The expression of a character takes place by formation of a specific protein. Transcription and translation say genes form specific protein. A gene is a segment of DNA. mRNA is formed on the DNA by transcription, and then mRNA helps in construction of a polypeptide chain by translation. So DNA forms mRNA by transcription, mRNA forms polypeptide chain of protein by translation. Thus, mRNA acts as an agent in conveying information from DNA to cytoplasm. mRNA is the messenger RNA. A sequence of three nucleotides on the mRNA is called as a codon. mRNA पे तीन न्यूक्लियोटाइड्स को कोडॉन कहते हैं. Each codon codes for a specific amino acid. So these were all the 8 characteristics of the genetic code. This was suggested in 1954 by George Gamow. Gamow suggested the genetic code while direct evidence was given by Crick in 1961. So who suggested the genetic code? George Gamow. Who gave direct evidence for the code in 1961 was Crick. And then Marshall Nirenberg, Henrik Mathai, and our very own Indian scientist, Dr. Hargobind Khorana, deciphered or cracked the complete genetic code. They cracked the entire genetic code and found out which codon codes for which amino acid. So, which codon codes for which amino acid, the entire genetic code was cracked by Nirenberg, Mathai, and Dr. Hargobind Khorana. They cracked the complete genetic code by using artificial mRNA templates and a cell-free system. तो उन्होंने आर्टिफिशियल एमआरएनए बनाया और सेल फ्री सिस्टम, एक अमाइनो एसिड का पूल कलेक्ट किया which is outside the cell, and mRNA is artificial. You know the sequences of codons, and उसको आर्टिफिशियल सिस्टम में डाल के एमआरएनए के ऊपर स्पेसिफिक अमाइनो एसिड्स अटैच हो जाते हैं. So you know the sequence of codons. It is an artificial mRNA. So they prepared an artificial mRNA, and artificial mRNA को एक cell free system में डाला. Means it's not a cell. It is just an outside medium. And they put 20 types of amino acids. And for AUG, what got attached? Methionine. For GGG, what got attached? I hope you remember. For GGG, glycine. For UUU, what got attached? For UUU, phenylalanine. For CCC, what got attached? Proline. So they found out each and every codon specified a specific amino acid. And for that, severe Ochoa enzyme, polynucleotide phosphorylase, was used. Normally, which enzyme helps in that attraction of codon to amino acid? Amino acid tRNA synthetase enzyme. And amino acid tRNA synthetase enzyme की जगह they had used something called the Severo-Ochoa enzyme, polynucleotide phosphorylase, which was helpful in polymerizing RNA with defined sequences in a template independent manner. So Severo-Ochoa enzyme की मदद से Nirenberg, Mathai, and Dr. Hargobind Khorana cracked the entire genetic code. Remember this, given in the NCRT, ये याद रखना. In the RNA, there are 4 types of nitrogen bases, AUGC, and 64 possible combinations of triplets called codons are possible. So सबसे पहले ये सोचना था, एक कोडॉन सिंगलेट है, डबलेट है, ट्रिपलेट है या क्वाड्रुपलेट है. If a codon had only one base, means every codon was only one nitrogen base, then the number of codons would be 4 raised to 1. That means only 4 codons which cannot code for 20 amino acids. So a codon cannot be singlet. U will code for one amino acid, C will code for one amino acid, A will code for one amino acid, G will code for one amino acid. So 20 amino acid के लिए एक nucleotide cannot be a codon. Can a codon be a doublet, two bases? Then it would form 4 raised to 2, that is 4 into 4, 16 types of codons. चार कम पड़ गए, चार कम पड़ गए, and 16 codons are not enough to code for 20 amino acids. So a codon has to be a triplet, and that means 4 raised to 3. 4 raised to 3, 4 into 4 into 4 is 64 codons. 64 codons are more than enough to code for 20 amino acids. 64 codons can code for 20 amino acids. And that is the reason a codon is a triplet. So 64 combinations of triplets are possible. Sir, which are these 64 combinations and what will they code for? 3 codons serve as termination codons and don't code for any amino acid. Do we remember UAA and UAG and UGA are stop codons or termination codons? They don't code for any amino acids, which means 64 minus 3, 61 codons are sense codons, and 3 are antisense or nonsense stop codons. 61 codons code for 20 amino acids. 61 codons code for 20 amino acids. तो on an average, अगर देखें 61 codons code for 20 amino acids, which means 3 codons code for 1 amino acid. 3 codons code for 1 amino acid. That will be approximate. So let us see which codons code for which amino acid. Sir, yahan you have first nitrogen base, yahan you have second nitrogen base, and yahan you have third nitrogen base. So, ye first, second, third. So that will be U, U, U. And then you have U, U, and C. U, U, C. And then you have U, U, and A. So you have U, U, A. And you have U, U, and G. So that is U, U, G. So these are the first 4 codons. And then you have UC series. UCU, UCC, UCA, UCG, UCU, UCC, UCA, UCG. And then you have UA series. UAU, UAC, UAA, UAG. So you have UAU, UAC, UAA, and UAG. Similarly, G series, UGU, UGC, UGA, and UGG. Similarly, you have C, CU, U, CUC, CUA, CUG. Similarly, CC series, CA series, CG series, and similarly, U से बनेगा 16 codons, C से बनेगा 16 codons, A से बनेगा 16 codons, G से बनेगा 16 codons. So a total of 64 codons are made. 64 codons, of which UAA, UAG, and UGA are stop codons. They don't code for any amino acid. And 61 codons code for 20 amino acids. Which are these? कुछ हमने किए थे. If you remember, UUU codes for phenylalanine. AUG codes for methionine. GGG codes for glycine. CCC codes for proline. ये कुछ एग्जाम्पल हमने लिए थे. Now the entire code, UUU and UUC code for phenylalanine. UUA, UUG, and as a matter of fact, CUU, CUC, CUA, CUG all code for leucine. Similarly, AUU, AUC, AUA code for isoleucine. AUG codes for methionine. GU series codes for valine. UC series codes for serine. CC series codes for proline. AC series codes for threonine. GC series codes for alanine. AUA, UAC code for tyrosine. UAA, UAG code for stop codon. CAU, CAC code for histamine. CAA, CAG code for glycine. AU, AAU, AAC code for aspartame. AAA, AAG code for lysine. GAU, GAC code for aspartic acid. GAA, GAG code for glutamine. UGU, UGC code for cysteine. UGA is a stop codon. UGG codes for tryptophan. CCCD codes for arginine. AGU, AGC code for serine, which by the way is the same serine which codes for UC series. So UCU also codes for serine and AGU, AGC also code for serine. So six codons code for serine. And you have CC, CG, 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 CG codes for arginine. AGA, AGG also codes for arginine. So arginine ke also liye we have six codon and GG series codes for glycine. So check it here. We have how many codons? 64. How many are stop codons? 3. So 64 minus 3, 61 codons code for 20 amino acids. So as we said, average hona chahiye 3. But it's not like that. For phenylalanine, there are 2. For leucine, there are 6. For isoleucine, there are 3. For methionine, only AUG. For valine, there are 4. For serine, there are 6. For proline, 4. Threonine, 4. Alanine, 4. Tyrosine, 2. And then histamine, 2. Glycine, 2. Aspartame 2, lysine 2, aspartic acid 2, glutamic acid 2, cysteine 2, tryptophan 1. So याद रखना AUG codes for methionine 1, UGG codes for tryptophan 1, only 1. And then arginine 6 and serine also 6 and glycine 4. So, sir, total 1, 2, or rather 1, 2, 3, 4. I'll just speak the names. Methionine 1, leucine 2, isoleucine 3, methionine 4, valine 5, serine 6, proline 7, threonine 8, alanine 9, tyrosine 10, histidine 11, glycine 12, aspartame 2, lysine 2, aspartic acid 2, glutamic acid 2, cysteine 2, tryptophan 1, arginine again 6, and say serine we are done already, and glycine 4. So how many amino acids? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, and 20 amino acids. 20 amino acids are coded by 61 codons. 20 amino acids by 61 codons. AUG, methionine, UGG, tryptophan, one codon codes for one specific amino acid. AUG codes for methionine and UGG codes for tryptophan. तो ये एक एक हो गया. And similarly, for phenylalanine 2, for tyrosine 2, for histamine 2, for glutamine 2, for aspartame 2, for lysine 2, for aspartic acid 2, for glutamic acid 2, for cysteine 2, for 2, and then for leucine, rather, let's start with it. Isoleucine 3, isoleucine 3, proline 4, threonine 4, alanine 4, and glycine 4. And for leucine 6, serine 6, and arginine 6. तो ऐसे total 61 codons code for 20 amino acids, and that is a beautiful dictionary which was coded by Dr. Hargobind Khorana with Nirenberg and Mathai. Nirenberg, Mathai, and Dr. Hargobind Khorana have cracked the entire genetic code. Beautifully done is this. Now, in early 1990, Bock established the 21st amino acid selenocysteine. And selenocysteine is not a standard. Standard is 20. Selenocysteine depends on the availability of the element selenium, and selenium se banta hai selenocysteine, so it becomes the 21st amino acid. Question is, who will code for selenocysteine? All the other codons have specific amino acid. So UGA, which is the stop codon, UGA, which is stop codon, codes for selenocysteine. UGA stop codon codes for selenocysteine, and that becomes the 21st amino acid. But ये सब एनिमल्स में नहीं होता है, कुछ एनिमल्स में ही होता है सेलिनोसिस्टीन. ये याद रखना. तो स्टैंडर्ड कितने अमीनो एसिड्स है? 20 standard amino acids. And what are the salient features of the genetic code? The genetic code is a triplet and commaless. There are no punctuation marks. So, sir, ये है sequence of codons, and how is it read? If it is AUG, GGG, CCC, UUU, so codon is read. How is it read, sir? It is read as first codon, second codon, third codon. AUG first codon, GGG second codon, CCC third codon. And accordingly, amino acids and tRNAs will come. Can you have AUG as the first codon, you skip G and make GGC as the second codon? नहीं, अगर आपने बीच में skip कर दिया और GGC second codon होगा, then the sequence of amino acid will change. Amino acid sequence change हो जाएगा. So you cannot skip a codon. Similarly, if it is AUG, GGG, and CCC, which is the first, second, third, AUG, GGG, CCC, you cannot skip a codon, a nucleotide, that will make the sequence incorrect. Similarly, if it is AUG, GGG, CCC, then you cannot have AUG as first codon and yahan GGG as second codon and include GGG G as a part of two codons. So sharing of a nucleotide is also not allowed. Otherwise, third codon instead of CCC will become GCC, and the sequence of amino acid will change. So a skipping of a nucleotide is not allowed. Skipping of a nucleotide is not allowed. It is read as AUG, GGG, CCC. Uh, First one, second one, third one. Skipping of a nucleotide is not allowed. Skipping is not allowed. It cannot be AUG, GGC. That is not allowed. And as a matter of fact, sharing of a nucleotide is also not allowed. AUG के बाद ये G दूसरे GG के साथ नहीं आ सकता है. Otherwise, third codon में GCC, the sequence will change. So, समझो you have AUG and so you have CCC and you have GGG. Then AUG के बाद CCC के बाद GGG. You cannot have AUG and then CCC की जगह CCG and skip a nucleotide. Otherwise, the sequence of amino acid will change. And you cannot have sharing of a nucleotide. So AUG, CCC and GGG के अंदर AUG is first codon, GCC becomes second codon. Then GCC will code for a different amino acid. So second codon has to be CCC. So neither is sharing allowed nor is skipping allowed. So the genetic code is triplet and comma-less. There are no punctuations. That means the triplets are arranged one after the other without any gap or any space. So no gap or space. In other words, skipping a nucleotide or sharing a nucleotide is not allowed. It is non-ambiguous, specific. Every codon will code for a particular amino acid. Every codon codes for its specific amino acid. UUU codes for phenylalanine, UCC codes for serine, AAC codes for aspartame, GGG codes for glycine. Specific. Every codon has a specific amino acid that is non-ambiguous. But there is an exception. AUG codes for methionine and GUG codes for valine. What does GUG normally code for? Valine. But if AUG is not available, then GUG will act as the start codon. Do you remember AUG ki jagah GUG can act as start codon? And it will code for methionine. So GUG, which normally codes for valine, will code for methionine. Check this here. What does GUG normally code for? GUG normally codes for valine and AUG codes for methionine. But समझो you have a sequence of codons in which AUG की जगह you have GUG and then GGG and then suppose you have again GUG. Then what will first GUG code for? Methionine instead of valine. And this GGG code for glycine. And next GUG will continue to code for its regular nitrogen amino acid valine. So what is first GUG coded for? Methionine. क्योंकि गोल्डन रूल ये है चाहे AUG हो या GUG हो, first amino acid is always methionine. So what will GUG code for? Methionine. What will next GUG code for? Valine. So GUG codes for methionine, next GUG codes for valine. So GUG, which normally codes for valine, if GUG acts as start codon, it acts on methionine. So that is an exception to the non-ambiguous rule. Similarly, UGA is the stop codon, but UGA may code for selenocysteine. So, UGA is the stop codon, may code for selenocysteine. So, these become the exceptions. AUG codes for methionine and GUG codes for valine. But if AUG is not available, then GUG becomes start codon and codes for methionine. So GUG can code for methionine as well as valine. And UGA, the stop codon, can act as a codon for selenocysteine. That is unambiguous होना चाहिए, specific होना चाहिए, but there are exceptions of AUG, GUG, and UGA. Then the genetic code is degenerate. As 61 codons are available for 20 amino acids, two or more codons specify the same amino acid. Hence, it is called degenerate. Now, this is exactly ulta. Check this here. UCC always codes for serine. UCC always codes for serine. UUU always codes for phenylalanine. Uh, AUG or GUG is for valine. GGG is for glycine. So that is specific. It is specific that every codon, CCC codes for proline, AUG codes for methionine, AUA codes for aspartame. That is specific. Every codon codes for a specific amino acid. Every codon codes for a specific amino acid. That is called non-ambiguous. But iska ulta. Every amino acid is coded by more than one codon. Every amino acid is coded by more than one codon. That is called as degenerate. That is called degenerate. So CCC codes for proline. That is specific or unambiguous. But proline is coded by CCU also, CCC also, CCA also, CCG also. And that means every amino acid is coded by more than one codon. That is called degenerate. So every codon, specific amino acid. That is unambiguous. Unambiguous. But every amino acid is coded by more than one codon. More than one codon. That is called degenerate. So every codon codes for specific amino acid. That is degenerate. Unambiguous. But every amino acid is coded by more than one codon. That is degenerate. And then polarity. Polarity is the important feature of genetic code. It is read only in the 5 to 3 direction. Whereas mRNA always read, mRNA is always read in the 5 to 3 direction. mRNA का रीडिंग हमेशा C5 to C3 direction में होता है. AUG and then GGG and then CCC and then UUU. तो ये हुआ tRNA ribosome, and on translocation it will move by one codon. And which direction will the movement take place from? C5 end to C3 end. So reading by amino acid tRNA synthetase enzyme is always from C5 end to C3 end. Amino acid tRNA synthetase will read the codon always from C5 end to C3 end. So that is the polarity. Translocation of ribosome. movement of ribosome will be from C5 end to C3 end. So that is the polarity. And the genetic code has start and stop codons. AUG acts as the start or initiation codon, and UAG called amber, UAA called occur, and UGA called opal serve as stop codons or termination codons. And the genetic code is universal, means these rules of protein synthesis are applicable for all, चाहे bacterial protein synthesis हो या animal protein synthesis हो, plant protein synthesis हो, सबके अंदर UUU will code for phenylalanine, सबके अंदर CCC will code for proline, सबके अंदर GGG will code for glycine. So this entire rule for protein synthesis is universal. Some exceptions are found. Mitochondria of yeast and in certain protozoa like mycoplasma. So mitochondria of yeast and protozoa like mycoplasma do not observe the rule of protein synthesis. They will have a different sequence of codons for different sequences of amino acids. Mitochondria of yeast and mycoplasma जैसे protozoa. So this was it, and we will just analyze now a very beautiful logic. Degeneracy of the code is many codons code for one amino acid. There are 61 codons which code for 20 amino acids. So 61 codons code for 20 amino acids. One would expect 61 types of tRNAs with a specific anticodon for specific codon. For 61 codons, there should be 61 anticodons. That means 61 types of tRNAs. But tRNAs are specific for amino acid. tRNA are specific for amino acid. Every amino acid is picked up by a specific tRNA. And amino acids are 20. And codons are 61. 61 codons. So now the question is, do we have 61 types of anticodons? 61 types of anticodons for 61 types of codons? Or do we have 20 types of tRNA for 20 types of amino acid? Because every tRNA has a specific amino acid. So for 20 amino acids, there should be 20 tRNAs. So for 20 amino acids, 20 tRNAs. But for 61 codons, there should be 61 anticodons. So tRNA अटक चुका है. Should it be 20 types of tRNAs for 20 types of amino acids or 61 types of anticodons for 61 types of codons? So who proved this and what is the real answer? tRNA is specific not for its codon anticodon but for its amino acid. So every tRNA will pick up one specific amino acid. So for specific amino acid, there are specific tRNA with specific anticodon. Let's see. This was suggested by Crick, something called as the Wobbles Hypothesis. Wobbles Hypothesis क्या होता है? जब इसे let's understand this. So according to the Wobbles hypothesis, as suggested by Crick, Wobbles hypothesis as proposed by Crick, what will happen? So, समझो translation हो रहा है, and you have GUU which comes as a codon, followed by GUC, followed by GUA, followed by GUG. So what does GU series code for? GU series codes for valine. GUG, GUA, GUC, GUU all code for valine. So now what will happen? Sir, समझो you have GUU as the codon. It is read by amino acid tRNA synthetase. Message goes in the amino acid pool, and a tRNA will pick up the amino acid valine and go and fit, and codon pairs with anticodon. So for codon GUU, which anticodon will come? CA and A. So G will pair with C, triplet. U will pair with A, doublet. U will pair with A, doublet. Perfect pairing. That is called perfect pairing. And then this tRNA goes out into the cytoplasm from the E site. Remember, it entered A site, it goes out through E site. And then the next codon comes, say GGG. And which amino acid is delivered? Glycine. And then, samjho you have GUC coming in. GUC also codes for valine. And valine होगा तो tRNA will pick up valine and fit into the ribosome. Abhi, which tRNA? The one with CAA anticodon picks up valine. CAA anticodon picks up valine. तो वेलिन के लिए कौन सा एंटीकोडन हो गया है? CAA. And it will go into the ribosome and fit into the A site. And fit into the A site. तो A site के ऊपर अब कौन सा tRNA आ गया है? For GUC you have CAA. तो G और C के बीच में ट्रिपल हाइड्रोजन बॉन्ड बनेगा. G और C के बीच में ट्रिपल हाइड्रोजन बॉन्ड. U and A के बीच में डबल हाइड्रोजन बॉन्ड. बट कैन C पेयर विद A? C और A के बीच में तो हाइड्रोजन बॉन्ड्स बनते ही नहीं है. तो G will pair with C, U will pair with A, but C will not pair with A. तो C and A पेयरिंग नहीं होगा. चलता है, चलता है. And next you have say UUU. उससे आया phenylalanine. And then you got say GUA. GUA also codes for valine. And which tRNA comes? CAA. CAA has become the tRNA for valine. And CA will come. Again, G will pair with C. U will pair with A. Can A pair with A? A cannot pair with A. No problem. Chalta hai. And then say CCC comes. And it brings proline. And then GUG comes. Same tRNA CAA comes. Can G pair with C? Yes. U pair with A? Yes. Can G pair with A? No. Chalta hai. Now what I mean by chalta hai? Because there are more number of codons, 61, and less number of anticodons and amino acids, 20. 61 codons and 20 amino acid, every tRNA has the ability to go and pair with more than one type of codon. Every tRNA has the ability to pair with more than one type of codon. Same tRNA, CAA, 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 CAA. Same tRNA, CAA, 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 CAA can pair with more than one codon, GUU, GUC, GUA, GUG. So more codons and one anticodon of the tRNA can pair, and that requires a perfect pairing between first and second nucleotide, GCUA, 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 इनके बीच में परफेक्ट पेयरिंग होना चाहिए. But third nucleotide U pairs with A may be specific, C pairs with A is wrong, A pairs with A, absolutely wrong. G pairs with A, absolutely wrong. So, only pairing between first and second nucleotide has to be specific. First and second nucleotide का pairing specific होना चाहिए. Third nucleotide may be specific or may not be specific. चलता है, and if it is not specific, if it is specific, it will pair. But if it is not specific, then the third pair will not form hydrogen bonds. Rather, क्या होगा? First nucleotide, यहां देखना, first nucleotide, second nucleotide के बीच में hydrogen bonds बन जाएंगे. Third के बीच में कॉम्प्लीमेंट्री पेयरिंग नहीं है, तो इट विल नॉट पेयर, इट विल नॉट पेयर, उनके बीच में हाइड्रोजन बॉन्ड्स नहीं बनेंगे, एंड द टू न्यूक्लियोटाइड्स विल कीप ऑन शेकिंग, wobbling, and shaking or wobbling is called as Wobbles hypothesis. तो यहां हुआ परफेक्ट पेयरिंग, तो बॉन्ड हो गया, यहां हो गया, यहां हो गया और यहां हो गया. सी ए और जी ए के बीच में हाइड्रोजन बॉन्ड्स नहीं बनेंगे, एंड दैट मीन्स दे विल कीप ऑन शेकिंग और वॉबलिंग. तो सी एन ए के बीच में, ए बीच में, जी ए के बीच में wobbling hoga, and that wobbling is called as wobble pairing, and that is called as Wobbles hypothesis. So for 61 codons, do we have 61 anticodons? No, we have less number of anticodons, less anticodons, and every anticodon has the ability to pair with more than one codon by a specific pairing of first and second nitrogen bases, but third nitrogen base may pair or may keep on wobbling, and that is called Wobbles hypothesis. Sir, ये क्यों ऐसा है? Sir, how much time does codon pair with anticodon? It comes in the A site. And A site पे आने के बाद A site पे tRNA आता है, and then the ribosome shifts and tRNA is at the P site, and from the P site amino acid goes to the next tRNA at the A site. Again it shifts, tRNA goes to the E site and tRNA exits in the E site. तो tRNA आया था A site पे, it will go from A site to P site and transfer amino acid to A site. And again on translocation, it will go to E site and tRNA will exit through the E site. So, how much is the time required for this? Less than a millisecond. 0.00001 सेकेंड के अंदर ये टीआरएनए आता है, अमीनो एसिड डिलीवर करता है, अमीनो एसिड ट्रांसफर हो जाता है और टीआरएनए एग्जिट हो जाता है. तो इतना मिली सेकेंड के लिए पेयरिंग करना है, तो फर्स्ट एंड सेकेंड नाइट्रोजन बेस of codon and anticodon provide enough stability. थर्ड पेयर हुआ तो चलेगा, नहीं पेयर हुआ तो उनके बीच में वॉबल होगा, बट वॉबलिंग चलता है is less than a millisecond. So this is allowed by nature and this will help in reducing the total number of tRNAs. आपको 61 कोडॉन के लिए 61 एंटीकोडॉन की जरूरत नहीं है, तो साइटोप्लाज्म में number of tRNAs will become reduced. So we help in economizing the number of tRNAs by this beautiful phenomenon called as Wobbles hypothesis proposed by Crick. And then we have mutations and the genetic code. So what are mutations? Effects of large deletions and rearrangement in the segment are easy to comprehend. It may result in loss or gain of a gene or a function. Say, for example, point mutation. A classical example of one point mutation is a change of a single base pair in the gene for beta globin chain of hemoglobin. एक हीमोग्लोबिन के अंदर एक जीन है, उसके अंदर समझो सीक्वेंस है CCC and codon would be GGG and उसके अंदर C की जगह आ गया A, तो कोडॉन विल बिकम GGG की जगह GUG. तो GGG would have coded for one amino acid, GUG will code for another amino acid. तो हम लोग करेंगे ये ऑल टुगेदर, एक न्यूक्लियोटाइड के बेस पेयर का चेंज हो गया, देन दैट इज कॉल्ड एज अ पॉइंट म्यूटेशन. उससे कोडो जीन में न्यूक्लियोटाइड बदल जाएगा, कोडॉन में न्यूक्लियोटाइड बदल जाएगा एंड अमीनो एसिड बदल जाएगा. तो we are going to do that, a change of a single base pair in the gene coding for beta globin chain. तो समझो ये है बीटा ग्लोबिन चेन. Beta globin chain, hemoglobin has got alpha and beta chain. Hemoglobin has got alpha and beta chain. तो उसमें बीटा चेन ऑफ हीमोग्लोबिन के अंदर एक न्यूक्लियोटाइड के म्यूटेशन से codon changes, and codon changes, so the amino acid will change and amino acid glutamate की जगह वेलिन आ जाता है, glutamate की जगह वेलिन आ जाता है, and that is called as a point mutation which results in a condition called as sickle cell anemia. तो उसे सिकल सेल अनीमिया होता है. आने वाले चैप्टर में मैं आपको ये सिखाने वाला हूं कि एक न्यूक्लियोटाइड में, एक कोडॉन में एक न्यूक्लियोटाइड बदल जाएगा, then that is called as a point mutation and will result in some severe disorder. So we are going to talk in detail in one of the lectures on sickle cell anemia. It's an example of one nucleotide mutation called as point mutation. And then we have, sir, समझो a sequence of codon. Ram has red cap and we do insertion of a letter. एक लेटर डालते हैं between HAS and red. Ram has red cap के बीच में B डाल दिया. So now the sequence will become Ram has and red cap की जगह इधर B आ गया, it will become DRE and then DCA and then P. So sequence of codons has changed by point mutation. Now suppose we insert two letters. तो B के साथ I भी ऐड कर दिया. Then now B के साथ I ऐड कर दिया. So the sequence will become Ram has BIR EDC AP etc. BIR EDC AP एक्सेट्रा। तो एक लेटर डाला था तो सीक्वेंस BREDCAP हो गया। अब दो लेटर डाला, इट बिकम BIREDCAPA something. And if we insert three letters, say big, then Ram has big will become a separate codon altogether, and red cap will remain the same. So, where is the actual problem? If we add one nucleotide, then BREDCA. If we add two nucleotides, then BIREDCAP. But if we add three nucleotide, then only one codon is added. तो extra amino acid आ जाएगा. बाकी red cap will remain red cap and will bring the original amino acid. So main फर्क कहां पड़ता है? एक न्यूक्लियोटाइड या दो न्यूक्लियोटाइड, उससे फर्क पड़ता है. The sequence of all the codons will change. But if we add three nucleotide, sequence of remaining amino acids will remain the same. Only an extra amino acid is added because of three nucleotides. And समझो deletion किया, Ram has red cap में से R निकाल दिया, R निकाल दिया, then it will become EDCAPA something. So that is point mutation. If we remove two nucleotide, Ram has red cap में से R और E दोनों निकाल दिया, then it will become DCAP etc. तो वो भी सीक्वेंस बदल गया. And समझो RED red ही निकाल दिया, then Ram has cap etc. means remaining sequence of codons will remain the same. So again, deletion of one R changes the sequence completely. Deletion of two R and E changes the sequence completely. But changing of three nucleotides, we deleted three nucleotides. Red ही निकाल दिया तो cap और उसके बाद के nucleotides will remain the same. So insertion or deletion of one or two bases changes the reading frame from the point of insertion or deletion. एक या दो बेस पेयर डाले तो रीडिंग फ्रेम कंप्लीटली डिफरेंट हो जाता है. एक या दो डाले तो रीडिंग फ्रेम बदल जाता है. एक या दो डिलीट किए तो रीडिंग फ्रेम बदल जाता है. But that is called point mutation. So point mutation is addition or deletion of one or two nucleotide, which will change the entire reading dictionary. Entire cryptogram mRNA will change. But if it is insertion or deletion of three or multiples of three are inserted or deleted, then one or multiple codons or one or multiple amino acids will change, but remaining reading frame remains unaltered from that point. समझो हमने राम हैज रेड कैप में तीन डाल दिए BIG, तो राम हैज big but red cap will remain the same. So एक amino acid आ गया, remaining sequence remains the same because there were three nucleotides inserted. Similarly, Ram has red cap में से Ram has red ही निकाल दिया, three nucleotide निकाल दिया. Then cap and remaining sequence remains the same, and that is three or multiples of three insert or delete means one or multiple amino acids are added or deleted and the remaining reading frame remains unaltered, and that is called as a frame shift insertion or frame shift deletion. तो एक या दो बेस पेयर से रीडिंग फ्रेम फ्रॉम द पॉइंट ऑफ इंसर्शन डिलीशन changes completely changes completely. That is called as point mutation. That is very dangerous. But if it is three or multiples of three are inserted or deleted, then one or multiple amino acids are added or deleted. But the reading frame remains unaltered. Reading frame remains unaltered. Big आया, red cap will remain same. Red निकल गया, cap etc. will remain the same. And that is called frame shift insertion or frame shift deletion or mutation. So we ended with it. Which is more dangerous? A point mutation of one or two, or a frame shift of three or multiples of three? Point deletion or addition of one or two changes the reading frame completely. That is more dangerous. Three or multiples of three added or deleted only adds amino acid or deletes amino acid. But remaining sequence of amino acid remains the same. That is called as frame shift insertion or frame shift deletion. That is the safer kind of mutation. Chalo. I hope you've understood this, all of you. Nice logic we have done. Please remember this very well. Ciao. Take care.